Hi, hi everyone. My name is Nikhil Singh, and this is my summation for AI for Science Hackathon. The title for my summation is Automatic Annotation of Protein Sequences. So, all enzymes are made of one or more chains of amino acids, which determine their structure, behavior, and interaction with other enzymes and molecules. That means it should be possible to predict the protein's function and behavior given just the amino acid sequence. And being able to quickly annotate enzyme or any protein sequence with a highly accurate model would have many applications, such as reduced manual labor or sometimes complete automation if the model is like very accurate and the risk is a bit low. So, so the this is an instance of the data that we are going to work on and going to use for training and testing our model. So this is the instance, and this instance belong to class 11. So there can be any number of classes or any number of uh, enzyme types, and this particular instance has been taken from creature 9. So for example, we have different type of creatures that we get the enzymes from. So we have around 8.5 lakhs protein sequences which are labeled, and they have a label associated with them, and around 2.5 lakhs protein sequences which we will test our model on to see how well the model is performing. And these are normally available in faster format, but this time it's available in string format. So we have a protein sequence in a string format, and we have a label for that, and we have a creature for that. Yeah. So I'll briefly go over the methodology that we have used. So the first part of the submitter system is feature generation. So each protein sequence gets embedded into a 1024 long vector, which contains the information about that particular sequence into that vector. So this vector can be used for various purposes, like visualization of a particular protein sequence in either 3D or 2D, and finding a similar sequence using cosine similarity or any other metric, or we can train machine learning algorithms on top of that for doing a custom task. So so we calculate these embeddings for each sequences in training and testing set. After we have the embeddings in place, we train a multi-layered perceptron neural network on top of these embeddings to do the classification. And all these intricate and the step-by-step approach has been mentioned in the notebooks that I have attached with my project on DevPost. So these are the following embedders and basically the techniques that we employed to see how good it is performing. So the first one is to cluster different enzyme types into different enzyme types. We can use simple sequence similarity. So for example, if there's a high overlap between two sequences, so for example, two sequences can be AGGT, SATT, and things like this. So if there's a high overlap between them, then they must, not necessarily, but most probably they'll be in the same enzyme type. So that was the first, but that didn't perform very well. And next were like these are the NLP techniques that were employed for uh, genome sequencing. Like we have genome sequence, and we have trained fast text, glow, sequence to vec, and other transformer models using that data, like a huge amount of data, so that those models have a semantic uh, explanation or information about that particular sequence. So we used all these techniques to embed our sequences. So these are the results. So fast text performed okay, fast text and glow performed okay, and the protein transformer models range from 90% to 93%, and the best performing model was ESM1B model that I'm going to explain further. So these are the results on test sets. So these are the results on 2.5 lakh enzyme types. So this is the embedder that we used. So the name of this embedder is evolutionary scale modeling. The embedder used in our model is a transformer protein language model developed by Facebook AI Research. It's a state-of-the-art general-purpose protein language model which can be used to predict structure, function, and other protein properties directly from individual sequences. It was trained on around 250 million protein sequences in an unsupervised way to inculcate the knowledge of the structure and alignment of various protein sequences. So this is the uh, embedded that we used, and this is like the final submitted system. So the first step of developing the system was to pre-process the data. So all these sequences had varying lengths. So some protein sequences can be say of around 250 characters, or and some can be say more than 20,000 or 10,000. So we need to have a common length in order to feed those sequences to the machine learning model. So we did some pre-processing, like making all the sequences of the same length by padding or thresholding. The exact number that we used could be found in the notebook that I was dashed. Then after the pre-processing, we have the embedding extraction. We use the same embedder that I described earlier in my presentation, that is the ESM1B, to extract embeddings for individual sequences. Then after we have these embeddings, we train a model on top of it. So that model trained for around 2 to 3 hours on Google Cloud Platform's AI Notebook service with 64 gigs of RAM. I use scikit-learn, so I couldn't use GPU to do that. But yeah, so it took around 3 to 4 hours. Then after that, we have a model, and we can use that model to annotate the sequences into 20 of the into either of the 20 types of enzymes. So this is just a demonstration, and I just use 20 classes, but we can extend it to, you say, whatever. You can adapt the domain by changing the training data, and we can retrain the model, and the model would adapt the domain like that. So this was basically it. So these are the tech stacks that I used. So I used Python to do the coding. I used Google Cloud Platform's AI Notebook service. I used scikit-learn to do the modeling. I did some things on Google Colab, and I finally put in all my code on Google Colab. And bioembeddings is the repository that I used to get the ESM1B embeddings. It made my process really very easy. And the sixth one is the basic scientific libraries like NumPy, Pandas, and all these. Thank you.